What is biotechnology? Biotechnology is the use of cellular and biomolecular process to develop technologies and products to help us improve our lives and health of our planet. A few of the ways that biotechnology helps us is by screening for genetic disorders, the Human Genome Project, electrophoresis, which is pretty much DNA fingerprinting, cloning, genetic engineering, and gene therapy. Screening for genetic disorders is pretty much taking a test during pregnancy that looks at the birth defects by removing a small amount of the fluid sac that surrounds the baby in the uterus and examining that for different disorders and diseases that could be inside of it. Okay, so next we're going to look at the Human Genome Project, which is an ongoing effort to analyze the human DNA sequence. It's useful in determining whether individuals may carry genes for genetic conditions and in developing gene therapy. Recently, scientists mapped over 20,500 genes on our chromosomes. Each person has the same gene sequence, but we are unique because our base pairs, which everyone has different pairs. So you're probably asking yourself, so how does the Human Genome Project work? It can be really confusing. Okay, so this right here is pretty much looking at the Human Genome Project itself and how scientists look at it to see different genetic conditions, the base pairs. Pretty much it goes inside and looks at the gene sequences to help understand what's your DNA. Okay, genome mapping is done by linkage, restriction cleavage, or DNA sequencing. Okay, so the basic gist of the genome mapping is a genetic map identifying the distance between mutations in terms of recombination frequencies, which is your DNA genes. So it's the genes inside of your DNA. Okay, so next is gel electrophoresis, which is really cool because it's what cops use to solve crimes, and what it pretty much is an agarose gel, which you put the DNA inside, which it negatively charges the DNA, and the big ones stay up the top while the small ones move through because it's easier to move through to the bottom. Okay, so just to help explain a little bit better, the process uses the agarose gel. It looks like jello, and then they put the DNA into the wells. The wells are at the top, as you can see in the photo up here. Then what they do is they run an electric current through the gel, and that electrotide is the electric current. Okay, you can see through the photo up on the top left. And then what happens is the larger fragments of DNA get caught up on the top because they cannot move through the gel as easy as the smaller DNA fragments. So then from there, the DNA was negatively charged by the electrotide, and then pretty much they can compare the different DNA, but it is not a DNA it's not an actual fingerprint, but it's the unique sequences of different DNA inside of every human body. Okay, so if you go back in your biotech little folder thing inside of your notebook, you're going to see this sheet right here, and here's a prime A1 example of what gel electrophoresis looks like because it shows the different DNA patterns and everything. And here it's showing how it's separating the DNA and not cutting it. To think that it's cutting the DNA, but it's actually separating the DNA from the big to small because the small can flow through the agarose gel easier than the larger ones, making it capable of separating the DNA. Okay, so next we're going to be talking about cloning, and this is probably the most fascinating thing out there because it's cloning different animals. And no, we're not going to be talking about these type of clones right here. We haven't made it to human cloning yet. We've only made it to animal cloning. Like Dolly from 1977, she was the first clone ever, and she's a pretty fascinating animal. Okay, so let's talk about the process of cloning. First, you're going to have the donor of the DNA. That gives the somatic cells, and then you're going to have an unfertilized egg, which you're going to have to remove the nucleus from that egg. So then the somatic cells and the unfertilized egg with no nucleus fuse together, and the nucleus from the somatic cells get inserted into the egg, which pretty much then mitosis forms the embryo inside of the cell, and then what you have to do is you have to put the embryo inside of the foster mother, okay? So then you have the foster mother, and then the embryo develops normally into the offspring from the foster mother. Bam. So just think of the possibilities that could be endless with cloning. I mean, we could have cloned humans, cloned animals. You know, if you lose your favorite animal, you could have them cloned. Pretty much, it's an endless possibility out there. Okay, so all in all, we should know about the screening for genetic disorders like the amniocentesis, looking for the birth defects during pregnancy, the Human Genome Project, which helps look for genetic conditions, the electrophoresis, which helps solve crimes by looking at the DNA fingerprinting, cloning, which is just awesome because the possibilities are endless. The genetic engineering, which that is pretty much just any type of biotechnology. And then to finish it off, we have the gene therapy, which gene therapy helps with genetic conditions. Thank you for watching our video.